Thank you for the introduction. So yeah, great to be here. Thank you for the invitation. So I'll talk about I start my lecture with an outline, introduction on biology and genomics and epidemiology of influenza. I'll talk about the available data and surveillance efforts that are conducted by World Health Organization, and why, as Arnie was just described, why influenza is such a great model system for studying evolution that actually teaches us really a lot about all other systems, including cancer. And then I'm going to talk about a method that we have designed for performing evolutionary predictions for influenza from 1 year to the next. And this project started as just a regular research project on how much you can do with available data, how good you can predict evolution, and this actually became after that it became part of the vaccine selection process. So we actually now use this method to, in collaboration with WHO, to assist the decisions and show what such predictive models, how these models can help. So I'm going to talk about this. And so it's a nice example of how a research project can have a translational impact. So Tom was already talking a lot about viruses and epidemiology. So influenza virus is posing a significant healthcare issue. So it is estimated that it infects from 5 to 15% of the human population every year, and it results in 500,000 deaths worldwide. And the fact that it comes back every year, this is attributed to the evolutionary escape of the viruses that are able to reinfect the hosts who become immune to the previously circulating viruses. And so this is transmitted, as Tom was saying, it's transmitting via from human to human via respiratory droplets and direct contact. So this is a closer view into the viral particle. The virus is this is a RNA, single-strand RNA virus. The genome is about 13,500 base pair long, and it has 8 genes that are positioned on 8 segments. So this is a figure that represents the influenza. So here inside we have these 8 segments. Right, they're called, and then at the surface of the cells there are 2 proteins, 2 important proteins presented. So this is hemagglutinin, HA, and neuraminidase. So these 2 surface proteins are very important from the evolutionary point of view because they are the ones that interact with the immune system. So hemagglutinin, in particular, is the one that is recognized and neutralized by antibodies. But it's also the function of hemagglutinin is to initiate infection. So this is the protein that attaches to the host to the host cell and initiates infection. While neuraminidase is also recognized by antibodies, but its primary function is to enable release of replicated viruses from the cell. So after the virus has entered, due to hemagglutinin, it replicates in the cell, and then it can leaves the cell because of with help of neuraminidase. So these 2 proteins are very important. And for hemagglutinin, to the extent that they actually use to name the virus types. So we, for instance, have influenza A, H1N1. That this means that hemagglutinin is like of type 1, and neuraminidase of type 1 as well. There's H3N2, H2N2, and so on. So this is how the viruses, how influenza gets this lineages gets their name. So in human we have now 4 seasonal influenza lineages circulating. So there's H1N1, H3N2, and there are also 2 B-type viruses, lineages, Yamagata and Victoria. So these types are usually like host-specific, and sometimes they can be transmitted between hosts. So here's a figure about like the different types. So so it shows how the transmission can go. So not every type can infect every host, and not every type can be transmitted between hosts. So we have a lot of infections coming from the birds, like from duck and chickens. So they actually are a reservoir for many different types, and they can occasionally infect hosts, and then we may have pandemics. But these are usually different. So these are usually different strains than those seasonal strains that circulate every year and against which we vaccinate every year. So just a closer view into this currently circulating strains. So we have, so they used to be influenza, the Spanish flu H1N1 that was circulating from 1918, and it's said to have come from like it was a transmission from avian source. So it was called the Spanish flu. Then we also there was the Asian flu that was circulating. So this is from 1957. That also was transmitted from birds. But then this Asian flu was actually replaced by H3N2, which is now in 1968. That also came from birds, and replaced H2N2. It's called Hong Kong flu, and this is actually the one that I'm going to talk about the most because it actually shows most of the evolutionary dynamics and adaptation. Yes.
Is there competition between the strains? I mean, if I have immunity to Asian flu and I've got partial immunity to Hong Kong flu, when you said replaced,
I think that's what is suggested, and so it happened again. Like between H1N1 in 2009, there was also an import from swine flu, and they replaced the previous 1918 Spanish flu. So there's actually some they tend to replace they replace each other.
These are pandemic changes, which really means that they're coming from other animals, and there's very little immunological competition because of that. Because they've been evolving in other animals for a long period of time separately. However, on a yearly basis, a regular flu, there will be overlap in the immune response. Not identity, but because there's mutations, but overlap. And what's making it much more complicated to figure out is that every one of our immune responses is slightly different than everybody else's, so that the population is a distribution of immune responses, and the virus is even a distribution of viruses. So when you ask the question, is there overlap and does it protect you, it will protect some of us, but not others of us. And
but you could imagine a pandemic in one stream reduces the transmission in another stream, sort of below replacement, right? Something like that.
These changes really are different viruses. This is the reason this happens, and the whole thing takes over. Is literally that virus has been isolated for 20 to 30 or 40 years in a pig or in a bird, right? Yeah. So it has evolved to be an optimal bird virus. and it comes into you, and you don't have anything.
Yep. So I'm going to talk more about a competition within, between strains within a lineage, but this is also an interesting observation, right? Okay, so just a closer look into hemagglutinin, which is this the protein on the surface. So this is necessary to initiate this entry of the virus to the host cell, and it's also a okay. So this is it has two domains, HA1 domain and HA2 domain. This HA2 domain is mainly creates the stem of the protein. This part there is the end of the protein, right? And then here is this receptor binding site, this is the site that attaches to the host cell. And apart from this, there are also epitope sites, which have been identified. So there's 5 epitope sites, A, B, C, D, and E, that are not the same as this receptor binding sites, but they are around there. And these are the sites that are targeted by the host antibody. So this is where they attach, and once they attach this, then the cell can no longer attach to the host cell. So it's not exactly in this part here where the receptor sites is. There would be not enough room for attachment for antibodies, but they are around, and once they attach around, they can they just prohibit.
So what's special about the epitope sites? Is it like a geometric or spatial?
Yeah, this is like spatial. They are. This is like exactly. So this has to do with spatial conformation. And as you can see, like some of those epitopes are actually far from this receptor binding sites, but they still are. Still, when you have an antibody binding over there, then it can prohibit infection, entry to the cell, and this is because it may change conformation of the protein. And these sites are also pretty well conserved. So this we actually know. So they are not. There's a lot of evolution on the sites, and I'm going to talk about it in detail because this is actually what is being recognized by antibodies, but the sites themselves don't move. They're really conserved. Okay, so this is another view on the same, just another figure that shows the hemagglutinin. These are the red part are the identifying the epitope sites, and this is a attached human antibody. So this is just another figure for that. Okay, so we have. So we know that influenza evolves a lot, acquires a lot of mutations, and it has to do so to escape the host immune response. And it does it by the 2 ways how it can do that. And one is by the process called antigenic drift, which means just by acquiring point mutations. And as Arnie was saying, there's the mutation rates in influenza are very high. There's on average 1 mutation per replication, and this happens just because the correction, the error correction by RNA polymerases is inefficient in single strand RNA viruses. And the second process by which it can evolve is by reassortment. So this means that if you have a infection of a cell with 2 different viruses, these viruses can exchange segments. So there's I showed you before there were 8 segments, and then what can happen during replication is that the segments will be mixed, and there will be new virus coming out of that. And this creates a huge it may create potentially can create a huge change, right? So a virus can acquire a new hemagglutinin protein that will be completely different from the ones that were circulating. So it will like just not as one mutation only like can make slighter changes. This can be a process that has much higher impact. And this reassortment can happen within a lineage, so you can have just different viruses from H3 and 2 that exchange segments, but it can also happen between lineages from different species, and this is how this, as I was showing you before, how these different new lineages were actually appearing, and they had huge fitness advantage over other lineages, and they were like taking over because they could really infect. Because the host population did not have immunity against those viruses. So now we can have a closer look into some sequence data and evolution within here. It's H3 and 2 lineage, so it's seasonal influenza. So as I was saying, so we have a lot of sequence data available. So this lineage appeared in 1968. So we by now have more than 50 years of evolutionary data. And as it was data collection and sequencing was sparse in early years, now we actually get about 10,000 new strains every season. So there's really a lot of data. I think so. We have like 60,000 sequences from hemagglutinin neuraminidase available. Here on this figure shows a reconstructed phylogeny of the hemagglutinin protein. So these dots here are strains that are collected from patients. And then we reconstruct the tree by a maximum likelihood method. So you find the most likely structure of how these viruses are related, and you can attribute then particular mutations that happen on the branches of the tree. So now when you this is a reconstructed tree. It starts in 1968. It goes until 2007 over here, but of course it can go on until now. And then strains from the same year are colored with the same. We use the same color. And what is particular about this tree it's very elongated, right? It's basically one-dimensional. So when you take strains from the same year, you see that their last common ancestor is on average 3 to 5 years back. So there's it's very constrained, and you don't see a lot of. I mean, the genetic diversity within one season is quite constrained. And the shape of this tree is really a signature of the fact that there's very strong selection acting on this virus. So that's why if there's diversity, so there's many strains are just being removed, and there's only the ones that are on the fittest are staying, and while the others are removed. So when you look closer into this different fitness effects and sequence evolution on the tree, you can find some patterns in evolution, and clearly there is many more mutations that are epitope mutations that get to be fixed and maintained in the viral population, while non-epitope mutations that are outside the epitope region are very often lost. So this means right? So this is the intuition behind that is that there's positive selection on the changes in the epitope regions because they enable host immune escape, while mutations that are elsewhere would probably just interfere with some basic protein function. Like they may like lead to loss of protein stability that will stop folding, or just perform other functions. So now when you look here, like we can like propose a quite like simple test for quantifying this effect, and so we can compute how many substitutions you have as compared to the number of mutations. And substitution is a mutation that at some point takes over in the population, so that everybody, all viruses have that. So in other words, this would be a mutation that is at some point like that you see that it ends up to be on the trunk of the tree. So now we can compute this number for synonymous mutations. So these are mutations that don't change amino acid structure, the amino acid codon. So, and we can assume that these are mutations that are not under selection. I'm going to have a little caveat to that on the next slide, but let's assume that on this for the test. And then we can compute the same ratio for non-epitope mutations and for the epitope mutations. And we really see that the ones outside the epitope are that you see substitutions much less often than in the SNAL case of synonymous mutations. It's by the factor of 1/3, and while the epitope mutations are enriched, so you see them 2.5 times more often than fixations of synonymous mutations. Okay, so. Yeah, and here is the other like effect, which is the caveat that I mentioned about synonymous mutations. This comes from work of Ben Greenbaum and Arnie and Raoul, among others here. So what they have done, I think that Ben was already mentioning this. Was talking about some of this work in his lecture on Monday. So what they have done is they have observed that there's a different CpG content in viruses that come straight from another species and enter new host, like here the human, and that the GC content of this genome is like changing gradually with time to remind this of the host. So here what we see is that so this is the total number of CpG sites in a viral genome, and here we have time. So we see that and then so we just take. These are just different strains that are being collected in those years. So the green dots, they show the distribution of the CpG sites in avian viruses. So you see that they are high over here. And then this is year 1918, where H1N1, the Spanish flu, entered the host. And you see that with time, it was losing CpG content. And similar thing has happened for the red ones. So this is H3N2. Yeah. So this is supposedly this is the effect of because those viruses want to resemble more the host cells, so they are not recognized by the innate immune system. Okay, so there's this effect, and then once again about reassortments. So as I was saying, so these reassortments happen when we have co-infection of the cell with 2 or more viruses, and they can exchange segments, and then at the end we have a reassorted strain. So now it has been observed that not all combinations are equally likely, and that there are certain biases in information of reassorted strains. So in this work, this work here, it was shown like from experiments that when you like cross different lineages or strains, you end up you end up with some combinations are just more likely than others, and some you never see. So there's some. So to some extent, there may be some epistatic effects between segments, so that you keep on having the same pairs together. And then another observation is that that reassortments like within a lineage are primarily under negative selection. So if you reconstruct so this is a reconstructed HA tree, and then you can map when reassortment with neuraminidase happened in those strains. And then you, again, like in this argument when I was showing evolution of or acquisition of mutations in non-epitope sites, you also see that most of these events happen on the periphery of the tree. So they are not, you rarely see them on the trunk and like leading to fixations. So, so this, yes? Of viral evolution in general? Well, some virus, like is it only in flu or? No, so not, but like some other viruses also have like recombination, like HIV just have regular recombination, which means that it can break the gene in parts. And but this is, so here like reassortment is really that you don't have any, right, there's no, the segments stay intact as they are, so you only exchange the segments. Okay. So another aspect of like epidemiology of influenza is that there are certain geographical and seasonal biases. So influenza epidemics is mainly observed, like it's observed in winters, and so these are different countries. They are sorted by latitude. So there's north and south, and you see, and these are temperate regions. So you see that influenza, and this is the the prevalence, number of cases. So this is winter, spring, summer, autumn, winter, spring, and so on. And so you see that there is high peak. There's a lot of cases in the winter on the northern hemisphere, and in winter on the southern hemisphere. And while in this tropical regions, temperate regions, you see actually cases that are observed all year long, but on smaller, like the smaller prevalence. So yeah.
The reason why, for example, in Spain and Tunisia, in Portugal, there's no cases, whereas in France they are, for example, for the left part.
Here or?
Yes.
Oh.
Spain, Portugal, Tunisia.
Okay, I would have to like
France,
yeah.
Romania, and so on, that have like two sides. Right?
Yeah, understand. Yeah. So I would have to actually look into the source of the figure, but it may be that sometimes you have I don't actually know from which year this was taken, but it's like the surveillance is changing from year to another. So it may be an issue of surveillance.
Anybody who is on the equator has flu all year round.
Yeah.
And anyone who's in the northern hemisphere has it in the winter,
Mm-hmm.
Right.
which is the southern
Yeah.
and the southern
Yeah.
hemisphere,
Right.
and vice versa.
Right, but the years that are like,
Yeah.
for example, half in the Clinton, so the British,
Yeah.
whereas Spain is just one.
So with this type of data, you always have to be careful because it depends on the reporting.
Yeah, exactly. So it's survive. Yeah.
So if Tunisia and Spain and other countries are different in terms of
Yeah.
the reporting that
Or
they're doing,
if the
you're
budget
gonna
is
Yeah.
fair,
Yeah, yeah.
you've got a problem.
Yeah, it's very much so. Yes. Okay, I think you were. Yeah, please. Yeah, I was gonna take over.
I was just wondering with this sort of seasonal pattern that's different between hemispheres, if there's sort of different strains that evolve separately in these different regions, the northern and southern hemispheres, and if there's some mixing then between them.
Strain, like you mean this H1N1, H3N2, or I mean, I'm gonna show a bit more into that, like that on the next slides. But in general, there's a lot of mixing of strains like within lineage because like people travel, and then you observe both H3. So this is a figure that aggregates all lineages, so like H1N1, H3N2, and B lineages. But you also see that some years H3N2 has more infections than H1, or H1 has more infections. So there are trends in that.
As a generalization, if you wanted to predict the strain that's coming this winter here, you'd look at Australia or New Zealand now. So as a generalization, what you're saying has some truth to it. The strains that are evolving in one place do show up in other places.
Yeah. Yeah, they do.
Does that generalization work the other way around?
Mm.
If I'm going to predict the flu in Australia, next Australian winter,
Yeah.
will the northern hemisphere summer?
Yeah, I mean, you look. Yeah, it's informative. So, I mean, it comes out from. I think I'll show this on this.
Could I get, this brings up an interesting question. In today's world, you could say that's because we travel a lot in airplanes, right? But this has been true forever, before airplanes. So how did it travel before? Yeah, birds. Right,
Well,
so migratory
are you
birds.
sure?
No, like how could I be sure?
Well, I mean, 100 years ago.
I don't know that. Yeah.
Of course they know the sense. Yeah. But then 500 years ago.
But you don't know.
No, I don't know. Of course I don't know. I'm not,
We don't have data.
But I do know something. For example, when the Greeks wrote about influenza, first of all, we're not sure it was influenza. It could be other respiratory disease.
Yeah.
But when the Greeks wrote about influenza, they said it came from China. It came from the east, right? It migrated from what they meant by the southern hemisphere, right? So there's been a long time of recorded history which fits this notion of the virus is moving to us. Now, you could ask the question, I don't know, nobody's asked the question yet, what's about the winter time that's so important that you only get the flu in the winter time? I mean,
No.
why not in the summer?
well, Peter
Yeah.
Felice actually did a really
Yeah.
cool experiment.
No, I'm not gonna. Yeah, because.
He took cages where he put guinea pigs that were susceptible to flu, and he would put one cage of infected guinea pigs next to another cage with uninfected guinea pigs, and then he controlled the temperature and humidity. And he showed that low temperature and low humidity favored the spread of flu from one cage to the other.
Yeah.
There are a lot
But-
of variables. Actually, could be a lot.
But yeah.
Usually with viruses, there are three places where infectious diseases are at high rates. The first is classrooms like this.
Yeah.
Look to your left, look to your right, look how close you're sitting
Yeah.
to each other. The second is the armed forces,
Yeah.
right? The third is jails. Those are the three places where infectious diseases spread at very high rates.
Yeah.
Right? So you're in one of the three right now.
Yeah. So yeah. So like back to the story. So you, but like you do see like a lot of transmission in these regions where humidity is higher. So but this is like supposedly is due to like that it happens not like via like direct contact and not by droplet transmissions. Or I don't know if it's well known. Okay, I had more questions and like you yeah. Okay, then over here, I'm just keeping order.
Very much related.
Yeah.
said that it's quite linear.
Yeah.
Is this a sort of averaging over all sort of hemispheres and all strains?
Yes, yes.
My question was then, like, if you sort of redo this north to south distribution and look at the trees, the structure of the trees there, are they more linear in these like regions where it's only being transmitted in winter, when there's a short environmental sort of a brief period in which it can exchange with the active, compared to regions where it's constantly present?
I think I'm gonna like show you on the next slide a comparison between lineages that may like address this question. I don't think there's like enough resolution to look at that, but I also like haven't looked myself, and like I mean you could like quantify if you this like shape of the tree and like and look at that, but I think that you have but you do have evidence that like you can actually you see how the viruses where they came from just by genetic similarity. You often see that you have like a virus from the US and the next like and that is very close to a virus in like somewhere in Europe or in Asia. So there's evidence just from this phylogeny reconstruction. There's evidence there's transmission. Okay.
Quick question, sorry for asking, but why China has all the years sporadic and it doesn't have any seasonal? Is there any reason, or is it again missing data on the?
It's probably missing data, but yeah, I can check that here. Okay, so yes.
I would assume that the summers in the northern countries is still not as hot as the temperature throughout the year in the tropics, but still this sporadic influenza throughout the years in the tropical countries, where there's almost nothing in summers in the northern countries. Do you know why is that?
Yeah, so I mean, so this is as Tom was saying that like I mean, there's like some hypothesis why that it like there's like some climate-related transmission effects, right? That you need humidity, or like that humidity is like that you need like low humidity for transmission, or and yeah.
By coughing, right?
Yeah.
Sneezing, coughing,
Breathing.
right?
Yeah.
And expectorating, right? In hot air, holds more water, holds more of the droplets. Transmission is more efficient,
Yeah.
less efficient. I don't know.
It's also like when you like people gather
More
and
efficient, but it isn't
yeah.
definitely less efficient.
So there are two factors, like the disease itself is worse off in higher temperatures, but the transmission is improved in
Yeah.
humid
Yeah.
and
Yeah. So I think there's many effects, and also like that like people if people go out or like when they are
It's the reason it's hot now.
yeah.
And one of the old timers is
Do you-
that in the wintertime people tend to be indoors and
Yeah.
closer to each other and exchanging.
Subways.
Yeah.
Is there any more question? No. Okay.
could it also have something to do with vaccination in
Uh,
northern countries that like there's almost nothing over there?
I don't. It's not. It's like vaccination levels are high in like say in the U.S. and Europe, but it's not. Yeah. Yes.
It's an imperfect representation,
Yeah.
but it gives you an outline.
Yeah, for a trend. Yes, that is. Like, there's
The
no
absence
reason.
of data is not necessarily meaning that there's nothing there.
Yeah. Okay, so maybe I just yeah. So these are some like just geographical distribution of like the sample virus, like how the viruses were sampled across the world. But this is used for. I just want to show you some trend that we were like shown in this publication by Trevor Bedford and all. So they reconstructed H3N2 tree, and then you can annotate every virus from like where it comes, and then what are you like supposed to learn from this figure? There's like a lot of mixing of this from different continents, and yeah, there's like pretty well mixed. Why when you look on say H1N1, you would actually see much more much more clustered, much more like many more geographical clusters. So that's, and you also like what you see here is also that there is that the shape of the tree is also like elongated, but it's less so than H3N2. And similar things happen for the two circulating B lineages. So in both cases, you see like this actually quite prominent clusters. So now what they've looked in this publication is that they also looked at the average, like the distribution of ages and of the infected hosts. And it so happens that H3N2, the median age is like 30, while for H1N1 and B lineages, it's less. So it's 20 or 16. And yes? Yeah, you can ask. Yes, please. Yeah.
It's about the previous speaker. Quick questions.
Yes.
One of them is related to what the previous speaker was saying, that there is always a quasi-species. They talk about quasi-species almost in each individual, right?
Yeah.
So how can you do an average of the mutation?
Yeah. So we completely we don't. We just look on the consensus sequence from a patient. So there is a lot on. There's evolution within the host and there's diversity. I'm not going to talk about that. So we actually assume that there is a dominating sequence, and so you have one sequence from a patient.
So. The quick question is, how come it's so linear, the tree or the evolution? Is it because the reassortment? Because the reassortment should introduce non-linearities there, right? Crossing branches.
So this is only one. So these trees are also for hemagglutinin tree only. So there's no reassortment here, right? So this is linear. And you occasionally see, and I was showing the tree before with hemagglutinin reassortment events mark on that, and they were mainly, I was showing that they were mainly peripheral on that. So, okay, so this was that, and then just an interesting correlation that was observed in this publication is that this distribution is also can be sort of explained by travel times and travel ages of people, so that you travel a lot between your area ferries and between and 60s, and this sort of is correlated with the infections of H3 and 2. There's also a model in this paper which I'm not going to show, but the hypothesis is that H3N2 is more mixed because it infects adults who travel more, and because of that, it also this evolutionary strategy is to evolve to be able to reinfect the same host multiple times, while H1N1 and B lineages where there's less adaptation observed, they actually focus on infecting younger hosts who haven't had influenza before. So you also see less adaptation and less of these ages. Okay, so this is some hypothesis to explain these patterns. Okay, so I'm gonna now talk about the vaccine selection process, and then I switch to our approach. Okay, so there's this surveillance effort going around the world. So this is map of the world, and the green regions are the ones that contribute to the surveillance efforts. And then there are 5 or 6 WHO collaborating centers that gather data from this national health centers who take data from patients, send it and then send it to this collaborating centers. And then so this vaccine production process is quite lengthy. So it starts. So it involves this continuous surveillance and testing of viruses. So the first viruses are being isolated in November and December, and then they are being characterized over this period of time. And then the strain selection is being decided in February. And this is, so we're talking about February of one winter, and then we pick the vaccine for the next winter, right? So this, in 2019, in February we picked the vaccine for that is then given to people in October, so like many months later. And the reason is that it has to be done so much ahead is just because the process of manufacturing the vaccine takes so long. So once the strain is being selected, then the like high growth reassortments are being tested, because like not every virus can actually grow in eggs and be a vaccine. So it takes the right strains have to be generated, and then so this really takes quite a lot of time until this vaccine can be given to people in October and November. So it's like a bit like, I'm not gonna go through all of this, but this is like just a bit closer insight, more details about it. So there's all year long surveillance and data collection efforts. So local health providers gather data from patients. Then they send this data to national influenza centers, and there's like 143 centers in 113 countries. So this is what from the countries were here, the ones in mark in green. They subtype. They perform. They like find like which subtypes of influenza these are, and then they send this. This data is being sent to this six centers. So there's a center in London, in Australia, in Japan, in China, in CDC, in Atlanta, and so on. And these centers perform like very detailed characterization of viruses. So like sequencing, antigenic characterization, cross-reactivity with past viruses, and so on. This data is being then shared publicly, the sequence data only. So, and also the epidemiological data. So the count of characterized viruses are being reported in databases, like there's a FluNet database, there's GISAID database where the sequences are being shared. This is publicly available for academic community. So there is, so once you have like proper academic email, you can subscribe to this database. And the antigenic characterization of the viruses is actually not being shared. So this is like some of this data is being published by individual centers, but it's not part of the culture to share this data. And then like this data is being analyzed, and this is actually like where some of this efforts and like some of this methods and like our efforts are also entered, that we like perform this predictive modeling on this data that are then like part of this are being looked into, and they may or may not influence the final decision what enters the vaccine. Okay, so now I'm gonna talk about this the method that we have designed in the next half hour. So the question that we wanted to answer was the following. So you have a influenza strain tree like reconstructed, and there's several coexisting clades that are antigenically different, which means that viruses from this red clade are not recognized, are novel with respect to the viruses from the orange clade. So this means that they can reinfect the host who had this virus before.
Question, is
yeah.
that symmetric? I can imagine that an orange clade doesn't recognize a green, but the green recognizes the orange.
It's not. It may like some. It's not always symmetric. So you see that some antibodies are protecting better, so this is not entirely symmetric. But in general, like, but it would be like a second order like correction. I think like in many models make assumptions that it goes the way, and then, but I also may talk about it a bit later. Yeah. So then the question is like, which of those clades will be the one that like will be dominating the next season, and so like what, or like in other words, like where will be the trunk of the tree, like when it's still will like keep on growing into the next year. So this is a question about some like fitness differences between the clades. So if we knew the fitness, which is the growth rate for or replication rate for those viruses, then we would be able to just project it into the future. So this is like an example. So if we have a clade alpha, and this is like sample, and we know its frequency, so like what is the fraction of the population that it infected in at time t. Then if we knew also the growth rate of this clade, then we could project it into the future, like with some time tau, say which may be like a year ahead. And we could predict the differences, how these two different clades will, what will be the frequencies of these clades in 1 year ahead. And then because this is, then once you collect more and more data, like the data from the next year, you can validate your predictions. Yes?
First, so how large is the error bar for clade 1 and clade 2?
For,
error bar. I mean, that's, the bar is the average, right? For all of the, different variants in the clade.
So no, so you have to do this, okay, you assume that that all those strains have similar fitness. So this is actually sort of, this is actually how we,
or is it just a schematic
it's a schematic picture, but so these are strains that we have identified to have the same antigenic properties. There are like other secondary effects that I'm going to talk about. They're like this. They are due to like non-epitope mutations that, but I'm going to talk about. So there's some variance, but it actually, if there's too much, you have still like you're able to split the clade, and like so the error bars are not dominating. So
why the growth rate is negative?
Why is it negative? So so it's a relative growth rate. So like it will never be because we're talking about frequencies now. So you subtract the average the average growth rate. So this is a difference to the average growth rate. So this of all of them, yes, okay. yes. Sorry, that's a good question. So because we're talking about right, so they compete, but we normalize it to 1. Okay, so this fitness effects are like the deterministic component of this evolution. There are also some like stochastic determinants. Some of that are like mutations and recombinations. So like imagine if you have like 4 different clades. So if you have mutation that they can actually like change frequency, right? So this different clades can travel and like become another ones, and so this would add stochasticity to this process. And there are also reproductive fluctuations called genetic drift, which means that like not every just by the fact that we are talking about discrete events of infections, or that some viruses have infected a host that like didn't leave home and like didn't transmit with the same rate as others. So you will have some random effects that will also add noise to those predictions. So in this model that I will talk about, we actually will not consider those stochastic effects. We'll like assume that they are even across all clades. So so we will not include them. So we only attempt to. Sorry, I'm just go back. We only attempt to come up with the right quantification of fitness effects, and includes and this will be what will define our model. Okay, so now, I will just switch to the model, and this is very much inspired by what I was telling you about before. So there are like these two different phenotypes, if you will, that we can take into account. One is antigenicity, which is immune escape of the virus from the immune recognition of by the host. Because so if a virus if a virus is recognized by antibodies, then it cannot it will not cannot replicate well and spread, so it decreases its fitness. Another thing is other functions here, like we will call it attributed all to stability of the protein. So mutations. So on one hand, mutations can lead to this immune escape, but on the other hand, they can destroy viral proteins or make them dysfunctional. So there's this two forces that we take into account. So and I was saying earlier, we will attribute this antigenicity phenotype to epitope mutations. So epitope mutations may, once they appear, they may decrease binding affinity of an antibody, so leading to gain of fitness, increase of fitness, while other mutations may increase folding energy of the protein, so it will generate a fitness cost for the virus. Okay, so now we can also propose a biophysical grounded model for that. So one hand, the binding of a virus with an antibody, this depends on the affinity between the two particles, and it's like a two-state thermodynamic model behind that. So there is some state here for which there's a non-specific binding, but and if you so this maybe you can have a state where a virus is bound by antibody, but then once it acquires mutations, it will lose the binding, so the affinity will go down with mutations. And here is a similar case for stability. So you have a protein that has some low free energy, but then with acquisition of mutations, the free energy will go higher, and at some point it will pass the threshold, and the protein will no longer be folding. And this is so what I mean here is that we model this by this Fermi function. By Fermi function, because these are not linear effects. You really acquire mutations, and you can get one mutation, two mutations, but then at some point it completely changes the phenotype. Yes?
Is this somehow additive in the number of mutations versus epistatic interaction?
Yeah. So here we no, I don't mean we do now. I'm talking about the phenotype, which is the affinity, and then, so we actually don't I'm giving this argument that but it can also be true for epistatic relations. So you can assume that different mutations can have different effects, but it's difficult to measure or infer from data. So actually we will make an assumption that these effects are additive.
Why are you using the Fermi statistics?
Why not? Why not? I don't. The two states, yeah. Yeah. Yeah, and yeah. So exactly. So you have one state, right? It's protein is bound. In this case, you have unbound protein, unbound virus, and bound, right? So and then you have transition between these two states. So this is, and then the same thing for folding. You can either have folded state or unfolded states.
Care more about how virulent
they're not?
No, so we don't. So this is like you're talking. It does not. There's like a different effect, right? So fitness is really just how well it replicates and can dominate, and the symptoms. I think you're talking more about symptoms and effect.
Is fitness measured by how many humans infected?
Yes. It's like expected number of like infected. Yeah, you can think about expected number of infected hosts, and yeah. Okay, so. So now it defines something like binding probability to an antibody. So folding probability. These quantities can be then translated into fitness effects. So this means, and this is like this is the intuition is simple. Is that a protein that is not bound will be more fit than the one that is bound. So it will just have some. So the fitness effect will have will be proportional to the binding probability with some coefficient here, which we'll actually get from fitting two data, like this how important this effect is, and similar for protein stability. So this also, I mean, he's also simple, right? So like if protein doesn't fold, then it's not fit, so it cannot infect others and replicate. Okay, so so this was some background intuition for this model. Like if we had this phenotypes of affinity and free energy measured, then we could proceed with this straight away. But we actually don't have this. Phenotypes are not easily measured, and we have to use some other data to infer those quantities. And for that, I show you how we can use genetic data to get the proxies for this affinities and differences in folding energy. Okay, so on one hand, like if you look on antigenicity, so say that you have a virus I circulating here, and then we want to know how well it is recognized by an antibody that was created in response to infection with previously circulating virus J. So this virus J is somewhere in the past on the tree, and now. Having sequence data, the best thing we can do is to measure sequence distance between the two. So if we measure, and this is what we attempted, we counted the number of epitope mutations that are between those two strains. So like we want to see how an antibody that is against this virus J is how well it is recognizing virus I, but we don't have this data in hand, and instead of that, we're just using the virus itself to give us some estimate. And this is what we're going to use. It's going to be like just number of mutations in the epitope region between the two viruses, and this we will use as a proxy for this affinity with a negative sign. So like the higher the distance, the lower affinity. The lower the distance, like if it's 0, then this means that it's the same. And mutations will decrease decrease affinity. Yes.
Sort of a technical question. What is the distance measure that you're using here?
It just it's called like Hamming distance, so it's just right number of mutations. Yeah. Okay. And then for the difference in free energy, we can do a similar thing, and it's also motivated by something that I was showing before, that this non-epitope mutations are very rare on the trunk of the tree. So we can assume that something that is like some last common ancestor of a strain that is on the trunk has good folding energy, and then like any mutation that happens on outside, that happens between this strain and that in the non-epitope region will increase the energy. And here we just we just simply count those mutations. So as you were saying, this is like purely like additive model, and we ignore at the moment like any epistatic relations where like two mutations can have like higher combined effect. Yes.
And could epitope mutations have free energy effects as well?
Yes, they could. Yes, they could. But we couldn't really like infed it, like because you like do it on the sequence data. So we assume that they don't, and like we assume that this epitope region is just very flexible and that you allow all mutations. But it's not. Yeah. But yeah, there's no reason why they couldn't. Yes.
For antigenicity, when you looked at the number of mutations that differ in order to make this proxy, were you able to know which mutations were non-synonymous, or did you just say that all mutations?
No, yeah, so yeah, that's a good question. We look on non-synonymous mutations, so the ones that change amino acid. Yes. Okay, so this is some proxy measure that we're going to use for this biophysical phenotypes. And there's just one more component that I didn't talk about yet, is that antigenicity is more complex. It's not that you have one antibody from the past, but actually the host population is a complex, consists of many cohorts, right? So patients have been infected with different strains, so there will be different antibodies that characterize them. So it's not at all interaction with one antibody, but there's many of them. And what you need to do here is to somehow infer what is this immune cohort structure. And then for each of these immune cohorts, find this binding, characterize these antibodies and find the binding affinity to your virus. And so again here, what we see is that we actually know, we may actually have some guess about what is the structure of the immune cohort, because we actually know the history of infections. So this tree of viruses that also shows which viruses were circulating and which viruses were infecting hosts. So we know which viruses were prevalent and what were the sizes of this, and how many hosts they have infected. And so because of that, we can attribute just infections with a given virus and sort of combine them into this immune cohorts. So now so these are every immune cohort is just antigenic clade on the tree. And then for each of those cohorts, we have the frequency at a given, at every time. So we have a frequency trajectory of this cohort. And then we can compute affinity of the cohort to a given virus. And for that, we can use this epitope Hamming distance that I was talking about. And then so at the end, so then we can use this and then just sum over. So and then this antigenicity component will be just summation over all immune cohorts, over all effects of the immune cohort, right? So this will be the size of the cohort and this antigenic fitness effect that comes from the cohort, and this will define the fitness of the virus. Okay, so I think this is all that enters this model. So we have two different phenotypes. One is this antigenicity, another is stability. And so this phenotype is the one that quantifies the adaptive part of evolution, and this one actually this is the conserved part, right? So you penalize mutations outside the non-epitope and outside the epitope region, but the whole fitness model will be just the summation of the two effects.
Yeah, I mean, you define, I asked before that you define the fitness as growth rate, right?
Mm-hmm.
So how does the antigenicity and stability translate to growth rate? That's a simple summation, but I mean, the antigenicity and stability might be a very complex function. Growth rate might be a very complex function of antigenicity and stability. It's not a simple sum. So
Yeah, so I mean, so this is all like a question of model fitting, right? So I mean, these models, these quantities have some parameters, right? The shape of the sigmoid of this like Fermi functions, where is the slope, and also the coefficients that multiply this effect. But then, so now the question is like, is it a minimal model that can explain observed evolution or not, or should you add more components? Like, is it good enough? And then, like, the only way to check is to do model fitting to data, and see if you like how successful you are.
Yeah, and related to that, I was going to ask, wouldn't you want them to be multiplied?
No, they are independent. Yeah.
Totally unstable.
Oh, this is in the exponent, right? Oh yeah, yeah. Yes. So that is in the exponent. Yeah. Okay. So. Right, yeah, so this is the fitness model I was like showing you before, the like function that you take the like frequency of the strain, and then it's e to this f, and this tells you you can project the frequency into the future. Okay, so now like some results on the past data. So we performed this on historical data like before 1993 2013, and we were measuring the true clade growth. So we computed these predictions for clades that were circulating this time, and this was the true clade growth, which is the ratio between you were taking frequency, say in year where you were starting the prediction and 1 year ahead, so you could see like how this changed, and so this was like measured fitness. And then you could do the same thing with prediction. So you would take the same thing is in the denominator, but here you take the predicted frequency. And then you can like see how well they correspond to each other, and actually see like quite a good correlation. And the trends actually are pretty well. You get the trends pretty well from there, and like in 93% of these cases, the clades that were growing were predicted to grow. And in 76% of the cases, the clades that were declining were also predicted to decline. There are also some like false positives and false negatives that are off the grid. Yes?
Comment. I have some background in the financial time series, particularly markets.
Yes.
Are you playing totally fair here? In the financial world, it's really important if you're backtesting that you don't use the future
Yes.
to build your model and predict the past. You make a lot of fake money.
Yeah. So we, I know. Yeah. Actually, like we did very careful. So like even like when reconstructing trees, we were for a model for predictions, we were using data only up to this year, and parameters were trained using data from the like preceding years and all that. Yeah. I mean, you always So like you have to be you have to be very careful, and like also with like this is what reviewers ask for. So there's so there is. But of course, there's like many ways. Clients, yes. There is. Yeah. Okay. So and then you can actually use the same thing. For performing model vaccine strain selection. So like because now like with a model like this, you have like a principled way for deciding for designing an objective criterion. And here what you can suggest as a vaccine criteria is you want to minimize the distance. They want to pick a vaccine strain from currently circulating viruses, because this is how it's being done. But you want to pick the one that is going to be the closest to the future population. And like as a distance, you can use like number of epitope mutations. So you just want to know, which one is going to be. So this is like the strategy over here. And then you can like having the data next year, you can see how far you were from this like optimal choice. And this we have done for our model predicted strains, and we compare it with the actual vaccines. So these are the black black line against the gray line, and like so we're showing this was actually quite promising. Of course, there are like many like caveats here with the real vaccine is that there's like many other effects when you like really are deciding to update or change a vaccine, and like not every strain can grow in eggs and be an actual vaccine. There's a lot of costs associated with changing a vaccine, so you really need to have a good evidence that there has been a new antigenic variant to update the vaccine. So I didn't say that, yeah. So the filled ones are the years when the vaccine was updated. Yeah, so this is actually, so vaccines, the real vaccines are not being updated every year. Sometimes there's a decision to keep the vaccine. Okay, so now, yes.
Yeah, technical question. I just want to have a rough understanding of how many parameters you have and how many data you have. So roughly, yeah.
So there's 2 parameters for like for the Fermi function, right? Fermi function and the coefficients. So there's, like, I mean, actually, the actual model had 4 parameters. So there is, yeah.
So the output after you feed in the data, I mean, after feeding the data, you got 4 values.
Yeah. So you have 4 parameters that you feed. Yes. Yeah.
Okay.
So the question is like then like how like if these parameters change between years and.
Data might be very noisy, right?
Yeah.
Yeah. So how many data you have? So are the parameters are well?
You don't have much data because I mean there's like a lot of sequences. You have like 60,000. I was saying there's 60,000 sequences, but your real data points are years. Like you predict from one season to the next. So you have.
So you. My question: are the parameters are well constrained by the data? I mean, does the data vary a lot? Yeah, because I'm working on the.
Yeah.
Basically, the confidence interval will cross 0. You don't know whether it's predicted effect.
Yeah. Yeah.
So do you encounter similar problems?
Yes, and like yes, like I think default answer is yes, but at the end, I think it wasn't like varying. Like so, like we're showing that we could actually train the data from like 1 year, say like using 4 years to the next. And there were, and like maybe it wasn't like so, like it wasn't like it was quite stable. Like you could have like some range of parameters and it was like pretty stable with that. But yeah. But yeah, it is always a problem. And this actually here we really don't have that many data points. Sometimes it's even better because sometimes we have like sometimes actually like you may like predict something that is like 2 years ahead, but because you have fluctuations and there's like clade maybe like for the next year is not gonna go up, but it goes up in next year. But I don't have like so much time, but actually I think I'll skip over like antigenic data expression, but I'll show you. Yeah, maybe like I just like show you like, okay, so just like briefly. So what we did after that was after the sequence model, we actually incorporating antigenic measurements in that measure like how well a titer, like you infect a ferret with a given virus, and then you see how well it protects against another virus. So this is like phenotypic data that can actually cover this epistatic effects better. So we after we started working with WHO collaborating centers, we got access to this data, and there was follow up different models how to handle this data that is sparse and noisy, and there's many missing measurements. So you have to infer what are to really measure distance between every 2 strains. There's quite some challenges, but then we incorporated this into the model, and this is actually what we are using now instead of the epitope sequence distance. And there's some example of this different how you can predict this frequency trajectories from one year to another. There's 2010, 2000, 11. So sometimes you see that like your fitness effects are maybe like you overestimate them, but you get the trends pretty right. Here is like the other way, but like you're like this goes like there's some like the trends are like okay. This is actually a good prediction from 2013, and yeah, another one from 2014. So these are the like examples that I have prepared. But then, what I wanted to like to answer your question now fast. We came up with some measure for like seeing how good these predictions are, and for this you can like compare your predicted frequencies to the frequencies to the true frequencies, and then use relative entropy between the two. So you compare these two distributions, and you can this would be the like a measure for assessing the similarity, and in this information gain measure, we also see how much evolution actually really happened. So you also compare the true frequencies after a year to the ones that were like we started with.
Yes. I'd like to make a comment. There could be a curious effect in which your success would affect your observation. Let's do a little thought experiment where you are extraordinarily effective. You predict the strain that would occur.
Yes.
You vaccinate most of the population, and now the strain can't spread, and you will hardly see it.
Yeah. But like, like fortunately, this
You made correct prediction.
Yes, exactly.
What would have happened.
Exactly.
You
could.
vaccinate people, and now the strain can't spread because everyone's
Yeah.
vaccinated against
So in principle, you could include it in the model, right? So
Yeah.
this would be an effect that you could.
It's really, I mean,
Yeah.
it'd be very hard to.
Yeah.
Very hard to check that actually.
That would be good if it happened.
Yeah, if it happened. Yeah.
But you might not know.
Yeah. So I think that the vaccine coverage is not high enough for this to happen. But yeah, and in principle, this could happen. Okay, so then so just like maybe like I'm just gonna like explain like the trend. But we see when we do these predictions, we see that we actually have some like information gain over like null predictions that would just like keep the strain that is like the most prevalent now. So this would be one vaccination strategy. This is what you pick, but we see that using the model actually improves over that. And this is this information gain, and we see that the information gain is positive it's an increasing function for about 400 days, and then it plateaus. So this is sort of like gives like some kind of intuition how good these are, and what happens, and like how far ahead this predictions can go. And the reason why it plateaus is that when we do these predictions, we only talk about the clades that are circulating now, but then there's all the stochastic effects of new mutations, right? So the new mutations that appear, and then they need to be incorporated into the picture. But this result here shows us that using this for vaccine selection, this is like the time span is actually is reasonable. Okay, I think I'm gonna actually finish at that. Yeah. So this is what I'm gonna finish with. So like, thank you so much. Yeah, I only had like some like insight into like showing how we do this prediction, but I think this is what I wanted to tell. Thank you.